The overall goal of this procedure is to establish proliferative tetraploid cells, free of a diploid population, from normal human fibroblasts.
This method can help answer key questions in the field of carcinogenesis research, such as how aneuploid cells develop in the early stages of carcinogenesis. The main advantage of this technique is that proliferative tetraploid cells free of a diploid population can be established by relatively simple procedures. While some strains become completely tetraploid by simple treatment with spindle poison democoursine, most need to be handled with the shake-off method during med treatment to eliminate a diploid population.
To begin the induction of tetraploidy without the shake-off method, passage approximately 5 × 10⁵ cultured cells into a 100 mm dish. Incubate the dish in a 5% carbon dioxide atmosphere at 37 degrees Celsius for 24 hours. After this, treat the exponentially growing cells with medium containing 0.1 micrograms per milliliter DC. Incubate in a 5% carbon dioxide atmosphere at 37 degrees Celsius for 4 days. Then, replace the DC-containing medium with drug-free medium. Incubate in 5% carbon dioxide at 37 degrees Celsius until proliferation resumes. To induce tetraploidy utilizing